Evening all. Thank you so much for being here with me. Tonight we are going to dive into scientific reasoning. That is the fourth in our science series for TEAS prep. So get comfortable. We are really trying to get our all-in-ones honed down to about an hour, a little over an hour. So generally they were like 2, and we're trying to get them a little more honed. So I don't anticipate we'll have a full 2 hours tonight. So just giving you the FYI on that. So before we get started, I do want to remind you of all the wonderful things we have here at Archer Review to help prepare you for your TEAS exam. As you know, we've got the free live webinar. You can take a look at that QR code to see the ones that are coming up. I know my colleague Tammy's got an English and reading one, I think tomorrow. So we've got a lot, and we have a ton of them coming up in March. And we have a new format in March that you might really also enjoy when it comes to like science and when it comes to English and reading. We're going to have back-to-back where we have a night of content and then a night of actual practice testing. So I think that is really exciting. So look for that in our free webinar series. I think that's something that is going to be a new, fresh approach, and I think you'll probably really love it. Notice in our second QR code that you can rewatch these webinars. So any that you missed, you can watch them, or you can watch them at your convenience. So access there. We also have a fantastic question bank where you can tailor it to the areas that you need help in. I think that's so beneficial, especially for the people who have taken the TEAS and maybe didn't quite get the score they hoped, and you get feedback from ATI as to where you need to grow. You can really tailor that question bank to the areas that you need to grow in. So I think that is amazing. I really love that about our question bank. The on-demand videos. The on-demand videos is the content at a much deeper level. That is going to be the content that is more superficial in our webinars that we're going to really get into the nitty-gritty on. So check out those on-demand videos. Also, as you know, we have tutoring available, and I'll show you who all our tutors are here in just a bit. And then we've got 2 bundled packages. So we've got a SurePass and a SurePass Pro, and I'm going to show you those as well. So the SurePass and the SurePass Pro, the SurePass, you're going to get our question bank, you're going to get on-demand videos, you're going to get targeted videos for specific goals and your objectives. And then the SurePass Pro includes tutoring in there. So like today I had a couple tutoring sessions, had one young lady who was amazing, and I feel like she is just a question or 2 away from passing. So great experiences, great packages here. Definitely check those out. As most of you probably already know, since you're seeing us in some capacity on maybe social media, if not on Zoom, we're on Instagram, TikTok, Facebook. You can email us, reach out, and we are happy to respond. Also, these pages that I'm going through of the products that we have available to you, I also will show you at the end. So you missed a QR code or you missed a link, no worries, you'll see those at the end. And then lastly, you can scan these QR codes for the tutors. So my wonderful colleague, Tammy, she's our English and reading person. I am coming at you tonight. I'm Shelley Harshberger. I am the Archer Review science person. And then there is Mr. Brandon Kraft. He is phenomenal in our math area. So feel free to scan those QR codes. And again, I'll show these at the end if you would like to get them then as well. So tonight, what are we going to talk about? We're going to look at scientific measurement and tools. We're going to look at how to evaluate scientific information, how to predict scientific relationships or relationships in nature, and then how best to interpret scientific research. So our first one is really just mastering scientific measurements and tools. And our learning objectives with this one is focusing on the metric system for one, because in the medical field and globally, the metric system is what we use. So while the US isn't as universal with metric system. It is everywhere else. And in the medical field, it's primary. So we do have to learn how to manipulate the metric system and how to work and be comfortable within a metric unit. We'll also look at how to focus on models such as diagrams, illustrations, and graphs. We'll look at the appropriate tools for measuring different things such as length, mass, or volume, and then the appropriate scale. So to kick it off, as I mentioned, the metric system, which is also known as the SI system, is the key system that we work with within the medical industry. And the reason that that is the system is because it is global. It is the system used in the medical field worldwide. And so this ensures that we have precision and consistency in our communication. I mean, communication is the key, and we want to make sure we're communicating within the appropriate system. So as I'm sure most of you know, they are going to use basic units and prefixes to adjust measurement size. So we're going to use gram for mass, meter for length, and then what we are familiar with is second for time. Now, volume actually has a calculation. With volume, we have to multiply length by width by height to determine the volume. And volume is something that we would use for, the unit would be liter. That would be the unit we would use for volume. So the base unit, if we take a close look at this chart, is always 1 within the metric system. And then we move either left in our chart to go larger or right in our chart to go down. And we're moving by a value of 10 at each point. So if I'm in our unit for length, if I'm at meter and I'm going up, our next unit of 10 is decameter, hectometer, kilometer. Now, if I started our base unit and we're going down, we're getting smaller, our value of 10 less is decimeter, centimeter, or millimeter. Now, we do have this silly little phrase that we can use to try to remember them all. So king stands for kilo, Henry for hecto, died for deca, bi for base, drinking for deci, chocolate for centi, and milk for milli. And that helps you put them in order from largest to smallest. Cute little phrase. I know that I had a friend in college who would sit there and repeat that to herself when we had to do the metric system. So I guess it worked. So what you will often be asked to do, both in the medical field and on the TEAS, is to convert these measurements. So for example, maybe we want to convert 5 meters to millimeters. And we remember that a meter is 1,000 millimeters. And so we just move the decimal place 3 places. So we go from meter 1, 2, 3, and there we get our millimeters. Okay, so if we have 5 meters, we've got 5,000 millimeters. And so that's how we do that. We can practice it too. So let's say, for example, we want to convert 6,000 kilograms to centigrams. So we want to get here. That's our goal. That's where we want to get. So how do we do that? We start with our 6. There's our 6 kilograms. Oops, sorry. That happens sometimes when I'm trying to do the pre-planned here. So we want kilogram. There's our 6. Then we're moving by 10. There's 60. We moved over 10. 600, moved over 10, and see how our decimal place is moving. We started with it here. Now we have it here. Now we have it here. 6,000 here. 60,000 here to get to our 600,000 in the centi. So 6 kilograms becomes 600,000 centigrams. So we're just moving that decimal place 1 time for every unit. So now when we think in terms of distance, I know here in the US, we think inches, right? But here with the metric system, we're going to measure in meters or millimeters or centimeter. And when we're measuring distance, we want to use a ruler or a meter stick. That would be the tool. And it would be what we would use to measure width, height, or length of an object. And remember that we always want to start from the 0 point on the tool. And I think that that's easy to forget. If you have a ruler such as this one right here that we have illustrated, notice that our 0 point is actually here. So you have to start measuring from that point. So you need to be aware of the tool. You need to look closely at that tool and see, where does my 0 start? Does it start in a little bit? Because if it starts in a little bit, you need to measure from there. And each of the markings on a meter stick is typically displayed as centimeters and millimeters. So notice here, this would be 1 centimeter. And the lines in between them are millimeters. So each of these little lines are millimeters. So this would be like 5 millimeters or half of a centimeter, okay? All right, so if we had a mark that was just after that 1, let me clean this up a little bit so we can see it a little bit better. If we had a mark right here that we were measuring, that would be 1.1 centimeters or 11 millimeters. Okay, so that's how you do that. Now, if we want to measure mass, there are 2 common types of balances that we would use to measure mass. There's that triple beam balance. And you know, I'll tell you, it seems old school, but if you've been to the doctor lately, really, when you get on the scales at the doctor, it's a triple beam balance that is weighing you. So it's not as old school as you might think. But what you will more commonly see in a medical profession is the electric balance. That is going to provide a digital readout. It's more commonly used. That mass is going to be measured in grams typically or kilograms if you're weighing like an individual or a larger specimen of something. Now, as I said earlier, when we're measuring a volume, and in this case, it's a volume of a solid, we have a calculation. And so volume of a solid, like such as a cube that's on our screen, we're going to measure the length times the width times the height. And that's what we see here. 10 centimeters by 10 centimeters by 10 centimeters ends up 1,000 cubic centimeters. And it, oops, I'm circling things that aren't displayed on your screen yet. My bad. There we go. And that ends up being 1,000 cubic centimeters. And there's our unit there, okay? Now, measuring the volume of a liquid is a little different than measuring, obviously, the volume of a solid, right? And so in that case, we generally use a graduated cylinder. That's what's displayed on the screen. And what's so important when measuring the volume of a liquid is that meniscus. So the meniscus is the curved surface of the liquid. This is our meniscus right here, this area right there. And so when we measure it, we have to measure it right along the bottom, the center of that curve, okay? And if we do that in this visual, remembering that probably every mark here we see by looking at our graduated cylinder, every mark here is 1, there's our 5, there's our 10, there's our 15, there's our 20. So we know we're over 20. We haven't gotten to 25 yet. Every mark is a 1. So there's 21. And we look a little closer. We're trying to get the bottom of our meniscus. And more than likely, it's probably about 21.6. Okay. And that's how you measure it, right at that bottom of that meniscus. Now, one of the things that's really important when it comes to using the metric system is really thinking through the right unit. Do you want to be in grams? Do you want to be in kilograms? Do you want to be in milliliters, millimeters? What do you want to be in? And so it's important to kind of wrap your head around, well, what would make sense? Well, we don't want a ton and ton and ton of zeros, right? So for example, if we take a look at my Great Dane here, which that is a picture of my puppy, by the way, that's Bella. So Bella, probably her average weight would be about 50 kilograms. Now, obviously, we could say 50,000 grams, but that's excessive. We don't want all of those zeros. So it makes more sense to say 50 kilograms. So we want to use the unit that doesn't overwhelm us with zeros, either on a large scale or a negative scale. We don't, or not negative scale, but a smaller scale. We also don't want something that's like 0.0001 if we can avoid it. We want to put it in a unit that matches the item and doesn't result with a ton of excessive zeros. So some things to keep in mind, milliliters are best for small liquids like we have in our dropper there. Millimeters would be good for tiny objects like our little ladybug down in the corner. Centimeters would work for medium-sized items like your fist, your heart, even for these books that are pictured here. Meters are going to be for larger measurements like measuring buildings. And kilometers or kilometers, that's going to be like measuring distances. So if you think about, if you're a runner or you know someone that is a runner, if let's say that they said they ran a 5K, that K stands for kilometers. And so you would think about, it's not meters that we put that in, we put that in kilometers. And so that gives you a kind of frame of reference on when to use kilometers. So we got some key takeaways for this section. As we know, the metric system or the SI system is going to be used to measure mass in grams, length in meters, time in seconds, and volume in liters. From large to small, we want to go from kilo, hecto, deca, one, deci, centi, and milli. And we've got that mnemonic, King Henry died by chocolate milk, drinking chocolate milk. Rulers and meter sticks are going to be used for length. Volumetric flasks are used for liquid volume and balances for mass. And then we want to be logical about selecting the appropriate units and bases for our objects. So millimeters for small items, kilograms for large masses, and kilometers or kilometers for long distances. All right, so here comes the time of the night where you get to participate. Just before I throw a couple questions out there, if you are in Zoom, your answers are only seen by me. So if you're unsure, you can take a chance. No one will see it but me. If you are streaming on any of our social medias, I will see your answers, as will everyone else. So enter at your own risk, but I do hope that you participate. With these questions, I give you 45 seconds, which is about expected when you're taking the TEAS. So here's our question, and I'll get a timer going for us. Which of the following values is appropriate for the height of a refrigerator? Is it 25 centimeters, or I'm sorry, is it 2.5 centimeters? Is it 2.5 meters? Is it 2.5 kilometers? Or is it 2.5 millimeters? Get those answers out on our chats. And I'm going to get a timer going for 45 seconds. Oh, I'm getting some answers already. You guys are on fire. Okay, I think most of you got this right. Our correct answer would be B, 2.5 meters. Remember that centimeters would be like the size of a bug that we would be measuring. Kilometers might be a distance that a new runner aimed to run. And millimeters would be teeny tiny, like a flea maybe. This might be a ladybug. This would be maybe a flea. This would be a distance to run. So 2.5 meters is our correct answer. That's our final answer. All right, one more. Which of the following is the meaning of the prefix milli? Is it 0.1, 0.01, 0.001, or 1,000? Got 45 seconds. Get those answers in there. I'll put this up to help. There's that mnemonic. Maybe anybody who's hedging, that'll help. Okay, I think almost everybody got this one. The correct answer is C, milli. That's our milk in chocolate milk. Remember, B is our base, so this would be 1. Okay, and so that would bring it down 10, 10, 10. Okay, all right, we are moving on. You guys are doing great tonight. So our next one that we're going to look at is how to use evidence and logic to evaluate scientific information. And this, I think, is so pivotal today when we are being just bombarded with social media and other news sites. And we have to really kind of be critical as to how to evaluate it. And so this might help. So in this one, we are going to look at drawing evidence and logical conclusions from charts, graphs, or text. We're going to look at cause and effect in a scientific explanation. We're going to apply evidence to support that explanation. And then we're going to use data to make informed conclusions. So one of the things that's so important for us to understand is that science uses what they would call empirical evidence. That means that the information is obtained through direct observation or experiment. It's not based on theory. It's not based on public opinion. It is, I directly observed this phenomenon or this event, or I did multiple experiments and these were always my results. And so scientists are going to collect, analyze, and draw conclusions, and they're going to focus it on large data sets. The more data they have, the better we can trust the outcome. And so the confidence really comes from larger data sets and if we can reproduce those results. Cause and effect relationships. That's really what scientists are aiming to answer is, does this particular situation cause this particular effect? And that's what they're aiming to answer. So for example, let's say that scientists conducted a clinical trial and they are testing new medication for lowering blood pressure. They're going to give one group a medication, one group of patients medication, and they're going to give another group a placebo. Now, I bet many of you are familiar with what a placebo is. A placebo, typically in a medical situation, is a sugar pill. It is a pill that does not have any of the active ingredient in it. And they use that then as a baseline to compare the group to that's actually receiving the medication. Okay. And in this situation, what they're looking for is they want to know if the new medication, the cause, is going to reduce the blood pressure. That would be the effect. So they're going to control all the other factors for their patients. They're going to make sure that their diets are level, their activity level is the same, they haven't changed any existing treatments, And otherwise, the groups would want them similar in age, similar in every consistency. That way, they can confidently attribute any blood pressure reduction to the medication itself. They'll also do this over and over with hopefully similar outcomes. And if that's the case, then they'll be able to confirm this cause and effect relationship. Now, I talked about several things there in that experiment. There's an independent variable. That's what's being changed. So, for example, in our blood pressure medication, the independent variable was the blood pressure medication itself. Okay? The dependent variable is going to be what happens to the group receiving that independent variable. Does their blood pressure decrease? That's what we're looking for. That's our dependent variable. Our experimental group, that is the group that's receiving the medication. The control group is the group that's going to get that placebo, that harmless inactivated substance. They're going to be our baseline to compare and see if the blood pressure medication actually did benefit the patient. Now, it's important for us as people reading about experiments or as nurses that get information from their patients, we want to be attentive to how was the experiment designed? Okay. Was there a large enough sample size? Did they do repeated trials? Those are the kind of things that will tell us if this is medication we can put our faith in. Did they make sure that all the conditions were the same except for the independent variable? Did they keep everything else constant, like their diet, activity level, existing treatments, and so on? Those are things that you will need to read as a critical eye to help your patients understand whether some medication or some diet pill or something they want to try has truly been studied thoroughly. We also want to avoid bias. Okay, bias is the whole reason that we add a placebo. Bias is trying to prevent any anticipation or effect on the outcome based on what the participants or the researchers expect to happen. You know how if you go to work someday and someone says, "Wow, did you sleep poorly last night? You don't look like you feel good." And by the end of the day, you don't feel good because you've got this in your brain that they're telling you don't feel good. So we can influence the way we feel sometimes by the way we think. And so we have to try to avoid that. And that's where placebos come in. And that's also where a double-blind study comes in. A double-blind study is a study where both the researchers and the participants do not know who's getting the placebo and who's getting the medication. And in that way, we can assure that nobody's influencing that outcome from a psychological standpoint or otherwise. So with that in mind, that's another thing you need to do when you're looking at experimental data is looking for bias. Were the participants randomly assigned? Was there blinding in the study? At least the participants should be blinded, but in best case scenario, it would be a double blind. Also look for, this one always gets me, potential conflicts of interest. If weight loss company ABC is doing their own research and they're selling medication ABC, are those results potentially biased? Yeah. So keep that in mind. Okay, so this section we zipped right through. So some key takeaways. Empirical evidence. That is what science bases its information on. Direct observation, experiments, not opinions, not theories, not public opinion. Data analysis. We want to make sure that we have large data sets and reproducible results. We want to recognize that when there's a study, we're really trying to answer a cause and effect relationship. That's what we're looking for. And in those experiments, then we've got key things to keep in mind. The independent variable, that's what's being changed, like a group receiving medication. The dependent variable is the outcome. Did the blood pressure decrease? The experimental group is the group that's receiving the treatment. The control group is the group that's the baseline. They're not getting a treatment, and in most cases, they're getting a placebo. Bias, that is any influence, any anticipated effect. And you need to look for bias, look for random participants, look for blinding, look for potential conflicts of interest. If the company that did the research is the company who made the medication, anticipate bias. Okay? All right, so we have a practice overview here. I'm going to give you a moment to read through that. I'm going to then give you some highlights, and then we've got a couple of questions that go with it. So just hang on there, and we will go from there once you've had an opportunity to read it. So give us a quick second there, and you'll do. Okay, so as I said, I wanted to go over some key things, and then we've got some questions that go with this. So in this research that's been conducted, again, it's on blood pressure medication. I stayed with the theme. We've got 4 participants. Notice that 3 of the participants received the medication. 1 of the participants got our placebo. Notice that we have a base blood pressure rate at 8 a.m. And then we have a base blood pressure rate 3 hours later. And patients 2, 3, and 4 received the medication, and patient 1 got the placebo. Okay. Now, the details here will not be in our next question, but the data will, for you to be able to draw some conclusions. So here's our question. Which of the following conclusions can be drawn from the data? A, blood pressure medication reduces patients' blood pressure after 3 hours. B, the blood pressure medication does not affect blood pressure levels. C, in large doses, the medication is effective. Or D, the correct time to take blood pressure medication is 9 a.m. You've got 45 seconds. Okay, so the answer they're going for here is A, based on the data. But I will say, and a couple people have drawn this to my attention, they're like, it's a pretty small sample size. Yes. And there's only 1 person receiving the placebo. Yes. That is great critical eye. You've learned something, right? So would we put all of our confidence in this medication with 4 patients? No. Would we say, looks like on this teeny tiny sample that maybe we've got something here, we need to explore it further? Absolutely. Okay. But I agree. Some of you are like, wait a minute, there's only 1 placebo patient. There's only 4 patients in total. And you're so right. Also, I've got some other questions out here, and this is a great time to answer them. One of the questions is there an easy way to differentiate between dependent and independent variable? And there is. Start with whatever's being changed. The independent variable is what is being changed. The dependent variable then is what is being measured. It is what the independent variable is trying to achieve. Like we talked about cause and effect earlier. Cause is our independent variable. It's the medication. The effect is hopefully it's lowering blood pressure. Then someone else said, would the people receiving the dependent variable also be the experimental group? If we said, would the people receiving the independent variable, then yes, they are the experimental group. But the dependent variable is the result, the decrease in blood pressure. That's the dependent variable. So that's a good question. So you guys are so smart. You are following along, and this is great. So let's move on. All right. So in this case, it's the same scenario, but I just wanted to refresh your brain in case you needed it. It's asking us which of the following describes what was done to try to avoid bias in the experiment. So A, all patients were given the medication when blood pressure levels were highest. B, all patients had similar starting blood pressure. C, a placebo was given to the patient who did not receive the medication. Or D, the experimenter followed the same directions each time when administering the medication. Get those answers in there. Okay, I think most of you got this. It's C. So the fact that they did do at least one placebo was an attempt. Was it enough? No, but was it an attempt? Yes. I did have a request to go over these. For A, all patients were given the medication when blood pressure levels were the highest. No, I mean, all patients were given the medication at 8 a.m. See that in our data table? That doesn't mean that's when their blood pressure was the highest. It was all at the same time. B, all patients had similar starting blood pressure levels. I mean, not perfectly. There's actually quite a difference between like 162 and 150. So I would say 162 is really dangerous. 150 is dangerous, but not nearly as bad. So I wouldn't say they're all very similar. D, the experimental followed the same directions each time when administering medication. Yeah, they did, but that doesn't reduce bias. And in that case, B doesn't reduce bias either. Yeah, maybe you could say they had similar starting blood pressures depending on your opinion, but that's not about reducing bias any more than they followed all the directions. Yeah, that's great that they did, but they didn't reduce bias. The question asked about reducing bias. And I think that's important when you have some of these lengthy read heavy questions to really be attentive to what are they asking. Even if some of the answers are right, what are they asking? And that can help you narrow it down. Okay. So our next category, we are looking at predicting relationships. And so in this case, we want to evaluate the size or magnitude of objects. We want to look again at those causal relationships. We want to look at sequence of events. And we also want to assess like the impact or scale of events. So when we're thinking in terms of magnitude of size, what's important for us to understand here is the appropriate tools and the appropriate methods for patient care. And I think examples are really what bring this one home. We wouldn't use the same tranquilizer dosage on an elephant that we would use on a cat, right? And so we've got to be attentive to magnitude of size there. We wouldn't use a scalpel to remove a femur, yet a scalp, or we would use a scalpel, my bad, to remove a femur, but we wouldn't use it to try to cut an animal in half. That wouldn't be possible. So we've got to be attentive to being logical. Does this make sense? Oftentimes, I think when you're taking an exam and you're kind of stressed, you got to really just stop, take a deep breath, and go, would this answer make sense? And I think that's pivotal there. Some more things to be attentive to is biological organization. So again, we see that metric system come into play here. And we pull in some of the more really small measurements. So we've got nanometer. That's going to be a billionth of a meter. That's going to be something that's measuring like an atom. We've got a micrometer. That's a millionth of a meter. That's going to be measuring a molecule as well as the nanometer. It depends on what molecule we're talking about. We've got the micrometer also measuring organelles within cells, and depending on the cells, sometimes even the cell itself. Then we've got millimeter. That's our thousandth of a meter that we talked about earlier. And millimeters or centimeters, a hundredth of a meter. That would be measuring like body tissue. Centimeter or decimeter, the tenth of a meter, could measure things like your fist, your heart. A meter would be the base unit, and that's going to be measuring like the entire organ system or the entire organism as a whole. And we also have another silly phrase for this. Now many millipedes crawl down mountains, stands for nanometers, micrometer, millimeter, centimeter, decimeter, meter. And so if you put that all together, that would help you get into those biological measurements that go really small up to the meter. All right, so we're going to practice, and this one's a little harder. You can't really put answers in the chat box as well, but you can follow along with me. So I'm going to give us about 30 seconds on this one, and then I will kind of slowly reveal each of them. But the idea is to match the structure to the correct unit. You've got 30 seconds. Okay. So our red blood cell, that's going to be micrometers or micrometers. You can say it either way. That's going to be pretty small on our scale. Human height, meters. Human heart, centimeters. Atom, about as small as you'll go when you're studying for TEAS, the nanometer. Human torso length, decimeter. Now, magnitude of processes. I think this one's also a little challenging as to, well, what do they really mean about that? And they want you to compare like biological processes. So if any of you remember cellular respiration, if you remember that the type of cellular respiration that does not require oxygen, as in anaerobic, that an in front of the word aerobic means no oxygen, that type of cellular respiration, or that first step, I should say, in cellular respiration, is glycolysis, and it only gives us 2 ATP. And remember, ATP is our body's fuel. It only gives us 2 ATP per glucose molecule. It's not very much. But if we have oxygen available, remember that's what aerobic means, then the results of glycolysis can go off into the mitochondria. And in total, we can end up with about 36 ATP. Notice that there is a huge magnitude of difference here. Glycolysis doesn't give us very many ATP. The whole process of aerobic respiration does. And so that's helping us understand that that's what they mean by magnitude of process. We want to see a vast difference between outcomes of things, and that would be a degree of extreme magnitude of difference. Now, causal relationships, we've talked about that a little bit. This is where the scientists are going to use data that they've gathered through experiments and observations and recording patterns and trends and outcomes over time to draw conclusions. And they're going to ensure that the sequence of events, that the cause occurs before the effect. They're going to make sure they're only manipulating one variable at a time. That's our independent variable. And they're going to conduct many, many trials to see if the same cause consistently produces the same effect. These are all examples of that. So we see in our world today that there's a cause and effect relationship between poor diet and obesity, lack of exercise and heart disease, exposure to UV radiation and skin cancer, smoking and lung cancer. And that's what we have here. And this is, another thing you need to be able to do with TEAS. You need to be able to read the graph. Notice here that cigarette smoke, that's our blue line. That's from the 1900s we got till about 2000. The black line is the lung cancer deaths. Notice that the per capita of cigarette consumption is on our y-axis. Okay, and notice there is a little bit of a delay. We start smoking cigarettes, do we die tomorrow? No, but we get lung cancer after a while. And as cigarette smoking increased, so did the lung cancer. Then as we start learning, oh, wait a minute, maybe cigarette smoking is not very good for us, then we start seeing that decrease. And that's what we're seeing here. Okay. All right, so another aspect of this idea of cause and effect is feedback loops. And you'll see feedback loops, especially in reference to homeostasis. And so there are positive feedback loops and negative feedback loops. And really the key difference between them is that positive feedback loops are going to amplify the change. Positive feedback loops amplify the change. Negative feedback loops, on the other hand, are going to get us back to a central place, back to a homeostasis. Okay, so we will look at those a little more closely. Someone had mentioned also that they thought the torso would be in centimeters on our matching. I think you could have put torso in centimeters, but since we were narrowing it down, the heart was smaller than the torso, so that made it better to do the larger unit for the torso and centimeters for the heart. But I agree that you could have used centimeters if you were given a choice, but since we were having to narrow it down between our options, that was the deal then. All right, so back to our feedback loops. So as I said, positive feedback loops are where we amplify some sort of situation. Childbirth is the prime example. So in positive feedback loops, we've got this situation where the baby is pressing on the cervix, it's causing tension in the cervix that activates stretch receptors, which send a signal out to the brain to release oxytocin. Oxytocin triggers uterine contractions, which causes more cervical tension. And this process keeps going on until the baby is born. Okay, so we amplify those contractions. We amplify that cervical tension. On the flip side, negative feedback is all about homeostasis. That is the thing with negative feedback. Think homeostasis. Okay. With negative feedback, we want to get our body temperature back to good. So we have this cold environment, our body temperature decreases, thermoreceptors, notice that change. That's compared to our 98.6 degree temperature. And that brings about muscle shivering, blood vessel constriction, getting our body temperature back to 98.6 degrees. Okay, so we're all about getting us back to homeostasis. All right, we got some key takeaways here. So remember that biology from simple to complex starts with atoms, which make up molecules, which make up organelles, which make up cells. Then like-minded cells will work together in their tissues. Similar tissues will work together to form their organs. Organs that have similar functions will work together to form our organ systems, and then all of the organ systems will work together to form the organism. Remember that those really small measurements within the scientific units or within the metric system, we can have this mnemonic. Now, many millipedes crawl down mountains, and that stands for nanometer, micrometer, millimeter, centimeter, decimeter, or meter. Remember that understanding these measurement skills is important for good patient care. Scientists are going to use sequence of events to ensure that the cause comes before the effect. That positive and negative feedback loops demonstrate causal relationships through sequence of events. And in that case, positive feedback amplifies, negative feedback counteracts the change, brings us back to homeostasis. All right, here we go. We've got a question. Which of the following illustrates a positive feedback loop rather than a negative feedback loop? A, insulin lowers blood glucose levels after eating. B, sweating cools the body down when overheated. C, heart rate decreases to reduce high blood pressure. Or D, oxytocin increases contractions during childbirth. You've got 45 seconds. Okay, I think almost everybody got this one right. Yeah. So remember, this is about amplifying a change. Okay. Positive amplifies that change. So that would be the contractions during childbirth. And I go over the others, but it might give us away on our next one. So let's take a look at our next one. Which of the following is an example of a negative feedback mechanism? A, platelets releasing chemicals to attract more platelets during bleeding. B, increase heart rate to pump more blood during exercise. C, sweating to lower body temperature when it rises. Or D, release the oxytocin during childbirth to increase contraction. You've got 45 seconds. So remember, for negative feedback, our key word there is homeostasis. Remember, we want to get back to a place of balance. And so with that in mind, C is the correct answer, because if we get hot, we're going to sweat to lower body temperature back to that 98.6 degrees. Okay. Each of these others were positive examples. So if you're bleeding, platelets will release chemicals to attract more platelets to clot so that you stop bleeding. If your heart rate increases to pump more blood during exercise, that's still continuing. It's amplifying the blood pumping. And then we already talked about oxytocin. That is an example of positive as well. Okay, guys, thanks for sticking with me. We've got one more to go. So like I said, we're honing these down a little bit as we transition to our new style next month. So I told you it wouldn't be nearly 2 hours. We'll probably be done, I'd say, about the next 15, 20 minutes or so. So in this case, we're really going to take what we've learned thus far and we're going to apply it. That's really the bottom line for this last category in TEAS is, can you take what you know about science, about the scientific method, and apply it? And in that way, they want you to be able to formulate a hypothesis, outline a basic experimental design, recognize those variables we talked about. So those of you that had questions about variables, we'll get to revisit those. And then we really want to evaluate, were the experimental results worthy? We've talked about that a great deal as well. So here is some alarming statistics. There was a study done in 2016, and it told us that only 28% of Americans can actually assess whether scientific information is credible or not. So whether you read it on social media, whether you read it on Google, Yahoo, whatever you use for your news source, they're saying that 28% of us can't really decipher that. So as someone who's in the medical field or going to be in the medical field, it's going to be your job to help your patients make sense of things. I kept using diet pills recently, and that's a prime example. A lot of people can spend a lot of money and not lose a pound. And so it's really kind of your job to help educate them. And so if you're going to help educate them, you need to know. And yes, someone told me that was very depressing, and you're right. It breaks my heart. So it's really going to be your job to be able to say, well, that's not a credible scientific research. And so that's why it's so important you understand all of this information we've been looking at tonight. So remember that scientists really begin research with a hypothesis. And I know in maybe even elementary school, we learned that it was an educated guess. But really, from a scientific standpoint, it's they've observed a phenomenon, they've done a little bit of research, and they've formed a hypothesis. And then they take that hypothesis and they test it, see if it's right or not. And they'll support it, meaning, yes, my hypothesis was correct, or they'll refute it, meaning my data says I'm wrong. And they're going to do that over and over and over with rigorous trials, repeated trials, rigorous peer review, and people will challenge their findings and all of that to come up with results that we can believe in. And so, as we talked about earlier, our variables are going to be the things that are either manipulated or held constant in the experiment. We know that our independent variable is what is changed. But here's something when we're thinking about looking at data that we do have to be aware of is that that independent variable, that thing that's changing, if it's put on a graph, is going to be on the x-axis. Now, the dependent variable, that outcome, so the independent variable, if we go back to our blood pressure medication group, the independent variable is the medication. The dependent variable is whether it decreased their blood pressure or not. That dependent variable, the decrease in blood pressure, if it was the case, that would go on our y-axis. And then all the other variables, we often call those constants, have to be kept the same so that we know if there's a change in our blood pressure, it's specifically related to that medication. If we change anything else, we don't know. Is it the blood pressure or is it because we started drinking more water? We don't know if we change things. So the best way that I can give you is a little glimpse into my life, and this is actually a real experiment. So these pictures here are orchids. These are known as Cattleya orchids, and I have grown them for many, many years. And I am a very busy person, and I'm maybe a little lazy when it comes to them, and I don't fertilize them very well. And I started noticing that they just really didn't flower as often as they used to. So notice how I had experience with it. I had real world observations. I didn't just randomly come up with an educated guess. But using my experience, using my observations, I hypothesized, I bet if I fertilize, my flower production will increase. So I made it a little more scientific. I said the addition of fertilizer will increase the number of flowers in my orchid plants. And I did an experimental setup. So I created two groups of these Cattleya orchids. So I had 20 orchids in total. I had my experimental group. They were the ones receiving the fertilizer. I had my control group. They were the ones not going to get any fertilizer. They both will still get their water and everything else, but they're just not getting fertilizer. So remember then, that makes my independent variable the fertilizer. So that kind of goes with a question that someone had earlier. Yes, the independent variable is what is being given to that experimental group. Okay. Now, my dependent variable, I often can say that my dependent variable is my outcome. Are we getting greater flowers? That's my outcome. That's my dependent variable. And then my constants are keeping every single thing else the same. So orchids don't like to be fertilized very often. So this experiment lasted about 8 months. So each application represented about once a month. And I collected data. So I collected data on the group with flowers, my experimental group, and my control group without fertilizer. And I had to kind of do some variation because, of course, flowers don't grow very often. So if we look at my visual here, orchid flowers grow out of what is known as a sheath. And so if I could even tell that there was a flower that was still down in this little sheath area, I considered that a quarter of a flower. If I had buds, as we see in this picture, and this is actually a picture of my orchid that was growing that day. If I had buds sticking out, I considered that 50% of a flower. If I could see what color the flowers are going to be, I made that 75% of a flower. Or if it was fully open, I made that a flower. So we can see we've got some data here, my experimental group versus my control group. And then we can see I've also put it on a graph. And so while maybe this data table here was meaningful, you can see that this is much more meaningful, right? We look back here and we can tell, yeah, by the 8th month, we got 4 versus 1 3/4. But here we can really see a difference. And so here we can see that our control group, yeah, they still flowered. I mean, they were not flowering before, they just weren't flowering as much. But on average, the group that started receiving the fertilizer, on average, had a lot more flowers. Okay. Now notice my independent variable was the fertilizer. That's what's along my x-axis. Notice number of flowers was my dependent variable. That's on my y-axis. Okay. So conclusion. Based on this data, the experimental group, the group with fertilizer, did produce more flowers than the control group. Those are the ones without the fertilizer. So it did support my hypothesis of adding fertilizer, increasing the number of flowers in Cattleya orchids. But of course, we've talked about this a lot tonight. Would it hold up under scientific scrutiny? No, it was one trial with 20 orchids. We would need many more trials. We would need many more orchids. But it serves as an easily understandable, nice, bright, colorful example of something I actually did. So I had a question. Someone asked controlled variables versus controlled group. That is such a great question. So let's go back here for just a quick second. So control group. Control group is the group that do not receive treatment. The controlled variables are also known as constants. That's all the extraneous stuff that we want to keep exactly the same for both groups so that they don't mess up our results, so that they don't impact our results. So where it says constants here, sometimes they call those controlled variables more than they do constants, to be honest with you. Okay, so that's a great question. That can be confusing for sure. Okay, I think I was even off the mark on time-wise. We're doing great here. Okay, so some key takeaways on this section of application. Remember, as one of you even noted how depressing it is, many Americans struggle to look at how credible or incredible, not incredible, uncredible, that's the word, uncredible scientific research is. As the first person that is going to interact with your patient as a nurse, you're going to need to help them decipher the difference there. And that's why it's so important to understand the scientific method and how research is conducted. Remember that research really starts with an observation. I observed my orchids weren't flowering enough, and so I formed a hypothesis, and then I performed a test. Remember that we've got key factors here. Independent variable, that's what we're changing. Dependent variable, what's measured or the outcome. Controlled variables are constants. Everything else is kept the same. Remember that the independent variable has to go on the x-axis when you're reporting any type of graphical data or when you're analyzing graphical data. And then the y-axis is our dependent variable. I remember that it is absolutely essential to trust in any research only after they've had multiple trials. It's been peer reviewed. That's important too. So I brought that up a little bit tonight. I didn't go in great detail, but that means where the research is turned in to experts, they scrutinize it and scrutinize it and scrutinize it. And then maybe if they're lucky, their research gets published. So it's quite a rigorous process. All right, so we got some questions. A scientist predicts that plants grown in sunlight will grow taller than plants grown in darkness. This prediction is known as a hypothesis, a scientific assumption, controlled variable, or constant. You've got 40 seconds. Okay, I think most of you got this one right. Well done. Our correct answer here is hypothesis. And someone had pointed out that the keyword here is predict, and I think they are 100% correct, right? A hypothesis is the scientist predicting what they think is going to happen. And they may find out they're wrong, right? But they're going to predict that. So that was great. Thanks for bringing that up. All right, we got a couple more. When plotting, oops, when plotting research findings on a graph, which variable goes on the x-axis? A, dependent variable, B, controlled variable, C, independent variable, or D, constant variable. We've got 45 seconds. Okay, I think almost everybody got this one correct. The x-axis is the independent variable. This is our x-axis. The dependent variable is our y-axis. Okay, and the other variables do not typically get graphed. All right, our final question of the night. Which of the following is true about the dependent variable? A, it remains constant during the experiment. B, it is the factor measured during an experiment. C, it is tightly controlled by the experimenter. Or D, all of the above. Got 45 seconds. Okay. It is B, the factor measured in the experiment, or as I said earlier, it's kind of the outcome. And someone else noted constant variable, if it's the same as the controlled variable, yes. We could say that constants and controlled variables are used almost synonymously in scientific research, as I've used it tonight. Okay, so let's show you a few things here and we'll be wrapping it up. First and foremost, as I promised at the beginning, there's all of our QR codes to access all of the wonderful things we have. And as I said, I think, don't hold me to it. My English and reading colleague is doing her free webinar. I believe it's tomorrow night. But you can get that from that first QR code. You can verify for me. You can rewatch webinars. You can access question bank, on-demand videos, tutoring, and then two of our packages there, the SurePass and the SurePass Pro. There is detail about the SurePass and the SurePass Pro, and I am going to send out the PDF of this, guys, so you'll be able to get these details that way as well. These are packages. The SurePass, you get the question bank, the on-demand videos, you're able to target the videos for your specific goals, and then the SurePass Pro adds tutoring to that package. That's kind of the difference between the two. And then as far as socials, you can get us on Instagram, TikTok, Facebook, email, and there are the QR codes for the different tutors. As you know, I am your science specialist, so that's me, Shelly Harshberger. And then here, I would love for you, while I'm pulling the PDFs together, for you to give me some feedback. Let me know what worked tonight, what didn't. And I got a few questions. Let's see. Someone's asking about a refrigerator question at the beginning. I don't know. I don't know a refrigerator question. I'm so sorry. But I am going to send out the PDF so you can get into the PDFs and find whatever one you were talking about. As far as the SurePass, that's a bundled package, and you'll also get that in the PDF. So at this point, oh, you're so welcome. At this point, what I'm going to do is I'm going to mute for just a minute. I'm going to get this all in a PDF and I'll put it in the chat box, both in social media and on Zoom. But I just got to pull that all together. So while I'm doing that, if you guys could work on doing the survey, I would be most appreciative. So I'll be back here with you momentarily. Thanks for your patience, guys. It's compressing right now so that I can share it with you. So just hang on. I am working on it. I haven't forgot you. All right, there you go, folks. Thanks for being such a great group. On my socials, you're next. All right, folks, thank you so much for being such a great group. And check back for March. Like I said, we have a new format where we have back to back. First night will be test prep, and the next night will be questions like you'd see on the TEAS. So looking forward to seeing all you guys. You have a wonderful night, and I'll see you again.